Hello students, so welcome back to the channel. In the previous video we have studied there are different types of pigments which are playing a role in photosynthesis. Major pigment is chlorophyll A, but other pigments are also present which we call as accessory pigment. Now we have see how these pigments are organized. We are finding they are organized to make a light harvesting complex. So light harvesting complex will have what? Light harvesting complex will have hundreds of pigment molecules which are attached to protein. It means light harvesting complex is having many pigments and these pigments are attached with protein. Clear? And these pigments, they are known as antenna molecules. Their work is to increase the efficiency of photosynthesis by absorbing some different wavelength. Then one reaction center is there. Okay, reaction center. So one chlorophyll A, only single chlorophyll A molecule is present in the reaction center. So photosystems are the one which have two things. They are having light harvesting complex and they are having reaction center. So see here in this diagram, please see, this is light harvesting complex and these purple color you can check, these are proteins where the green color pigments are present. So pigments bound to protein will make light harvesting complex. And see, this is your reaction center complex, which is having single molecule of chlorophyll A, because this is the one which is going to do the reaction. Now we are calling the two type of photosystems are there, and this classification is based on the order of the discovery. First, photosystem 1 was discovered, then photosystem 2 was discovered. And we are finding that these two photosystems, they differ with the reaction center. So in the photosystem 1, the reaction center, that is chlorophyll A, is having absorption at 700 nanometer. But in PS2, we are finding it is having the absorption at 680 nanometer. So we can write PS1 reaction center 700, light harvesting complex means light harvesting pigment attached to protein. Same for PS2. So I think so this slide is easy, beta. Now see next. Right now I told you that they are named according to the order of discovery, in the sequence of their discovery and not in the sequence of their function during light reaction. In the light reaction, first PS2 is used and then PS1. But first discovery was done of PS1 and then it was discovery was done of PS2. Who has done? These are the two scientists, Emerson and Arnold, and they were working on green algae chlorella. So let me assume it, just imagine this is a diagram in which the green algae is present and this green algae is actually chlorella. So what we are doing it, we are now only putting the light which is far red light, means whose wavelength is more than the visible wavelength, means nearly more than 700 we can assume it. Okay, so this light was falling it and we are finding oxygen bubble has stopped coming, means yield is decreasing. So this is known as red drop. The red drop is the one which was first identified. And it is when the light is of more than, or you can say nearly 700 wavelength. Now suddenly another light came, visible light with the torch light. This is a visible light. And visible light means less than 700 PAR region. Okay, beta. So what happened after the visible light falling? Oxygen bubble increased. So when oxygen bubble increase, it means there are two types of reaction center. One reaction center which can absorb wavelength more than 700. That is why we were saying P700 photosystem 1. Another which is able to absorb wavelength less than 700, we are saying P680, that is photosystem 2. The photosystem 1 is having absorption at 700 nanometer and photosystem 2 is having absorption at 680 nanometer. Second discovery was known as immersion enhancement effect. So red drop, please see here, it is a minimum photosynthetic yield when monochromatic wave of this much is provided. Enhancement when both are together. Is it clear? Now, simple logic, try to understand. In the photosystem 1, oxygen bubble stopped. Photosystem II, oxygen bubble came. It means photosystem II is responsible for photolysis of water during light reaction. Is it clear? Now see location beta. Understand the location. First, I'll try to explain the diagram here. See this. This is the grana. I'm drawing the stacks of thylakoid. I've taken suppose 3 stacks. So this is non-oppressed region. Clear beta, non-oppressed region, and this is also non-oppressed region. This region, this is oppressed region. So oppressed region is the one which is having the membrane on both the side. So this side will be lumen, this side is lumen, this side is lumen. PS2, please listen carefully, PS2 will be located in the oppressed region of grana. Why? Because PS2 has to do photolysis of water, and when photolysis of water is done, oxygen is evolved and hydrogen is split as proton and electron. Proton has to be accumulated inside the lumen. Understood? To accumulate proton within the lumen, PS2 should be present in the oppressed region of grana. Clear? And you have studied in the chloroplast structure. This is again grana and this is stroma lamella. So this stroma lamella is also not having any oppressed region. It means stroma lamella will also lack PS2. So please see PS2 is located in the oppressed region of grana. Its reaction center is 680. It is the one which is going to do the photolysis of water, means it is removing the hydrogen, or you can say it is separating the oxygen from the hydrogen. Is it clear? PS1 is the one which can present anywhere. It is present everywhere. Is it clear? It's not restricted. Means it can present in the oppressed region as well as in non-oppressed region of grana. It can present in stroma lamella. So it is located in the non-oppressed region of grana and stroma lamella. Its reaction center is more than 700. You can say it is the one which is reducing nature it is having. And it participates in both cyclic and non-cyclic. We'll be discussing in the next video what is cyclic and non-cyclic. Is it clear? So you can see here, check beta, this is your PS2. This diagram is PS2. The PS2 you will find in the oppressed region. Check. PS2 you will find in the oppressed region. Is it clear? And PS1 you will find in the non-oppressed region. So check, this is PS1 present in the non-oppressed region. And if you see in the non-oppressed region, you will not see PS2. Is it clear? The PS2 is located in the oppressed region and PS1 is located in the non-oppressed region. Up till here, you are comfortable? Based on this, let us discuss few MCQ. See, red drop discovered by immersion is due to disruption of photochemical activity of which one? It is because of the PS2. Is it clear? PS2 is not working, only PS1 was working. That is why we found red drop. In which of the following the rate of photosynthesis is decreased? It is decreased when the light is more than 680 nanometer. Two photopigment system means photosystem 1 and photosystem 2 was proposed by, answer is immersion and Arnold, not Arnon. It is Arnold. So I hope so this video has helped you to understand what are the types of the photosystem, how are they discovered, and where are they located in the chloroplast, in grana. Thanks for watching. I hope this video might be helpful for you.